10. Okay, we got this because it's only 2 chapters. Okay, so here we go. So I need to get my brain in this mode, and then I'm going to get rolling here. So are you ready? Okay. So multiple choice, there's like 43 questions because it's only 2 chapters, right? The last one I think had close to 60, but that was more chapters, right?
How many questions?
About 43, okay? So, you know, you're obviously going to have questions from 7, you're going to have questions from 6. Some are going to be straightforward, some are going to make you think just like the last test, right? And if you know your stuff, you should be fine, okay? So, here we go. Structure of the cell membrane. All right, so you have the phospholipids, right? And then embedded in the phospholipids, you have this. Yeah, what kind of protein is that?
Cholesterol.
Okay, there's a lot of right answers I'm hearing, but not cholesterol. It's in the membrane.
Protein.
Embedded. Interior.
Integrated. Integral, integrated, it's in, right? If this protein were on the surface, it's on the peripheral protein, okay? Inside the membrane,
What's the outside one? Wait, which one's the
That's the cholesterol.
okay, peripherals on the periphery, right? Integral is on the inside. There's your cholesterol, right? Then if we have this, and I ran out of space up here, but what if this was a chain of sugars like this, kind of coming off? What do the sugars, the carbohydrates do? Okay, think of blood cells and antigens. Like here's a blood cell, that's an antigen.
What's antigen? Protection.
That's the same thing, except that's more detailed, that's more basic. They're identifiers, right? So this helps your cells identify things. Okay, that's what the carbohydrates do. Okay, they're for recognizing. They're like antennas, and they kind of look like antennas in a way. You know, they just kind of come off, and it's for recognizing things. Okay, that's the basic makeup. That's what I want you to know about it. Okay. When you look at properties of the membrane, okay, so what region is hydrophobic? Right, so this part is the hydrophobic, right? And then the heads are philic, right? So the outside, or no, the inside and the outside are philic. Those phosphate heads, right? Okay, a couple more things with properties. How can this protein be embedded in a membrane that's made of lipids? What do we know about proteins and amino acids? What property do the amino acids have in this region?
Oh,
They're hydrophobic aminos. And then the amino acids in this region and this region must be hydrophilic. That's why it can embed itself. Okay. What purpose does cholesterol serve?
preventing the possible.
Yeah, they keep it fluid. Okay, because remember, it's a fluid bilayer. It's kind of waving, like ocean waves, right? And they just kind of stay fluid. So if you're a hibernating bear, is it a wise choice for your body to naturally increase your cholesterol?
Yes.
Yeah, because what does it do to your cold cells? Keeps them fluid. Because you're hibernating, you still need to move stuff in and out, okay? Now, if you talk about plants like winter wheat, when do we grow winter wheat?
In the winter.
In the winter, okay? So, which phospholipid does winter wheat contain? Right here, right? Because remember the phospholipids that have bens in them? What kind of fats are those? That's unsaturated. And what do we know about unsaturated? Can you put a bottle of vegetable oil in the refrigerator and it not become hardened?
Yeah.
It won't, because it's unsaturated fats. But if I take a stick of butter, which would be like this one, and put it in the refrigerator, and then I get it out and try to, how's that gonna go? No, it's gonna be rocks that you can't even, you ever try to put butter on bread and the butter was rock solid?
Yeah.
It's just, it doesn't work, right? Okay, so winter wheat has unsaturated fats, again, just like the bear, to help do what? Keep it fluid, so then the winter wheat can live in the winter time. Got it? Okay, that's that whole story right there, okay? That's like the first part of the homework from chapter 7. Those are kind of questions I was asking, right? Protein structure and types. Okay, we kind of already hit this already, right? So this one is an integral, right? And that one is a peripheral. Okay, integral proteins primarily do what to substances? Yeah, high to low, move stuff across, whatever, okay? Okay, so that's, those first three parts, that's all membrane background. The rest is all movement, okay? Can I move on? Okay, I'm gonna clear it. Okay, so now let's take membranes and let's kind of do a little table here. Okay, so we have passive and we have active. Okay, passive moves things from high to low. Active moves things from low to high, right? Types of passive transport. Diffusion. Right here, right? Diffusion, osmosis, and facilitated. Okay? So that's kind of basic stuff, just info. And then we get into active, pumps, co-transport, ion-gated, endo and exo, cytosis. Okay? Active transport works because it uses what? To move things across, right? It uses ATP, okay? Now, of course, I could list these things, but I'm gonna take the time to write them. But, I could list these here for passive and these here for active, okay? But now let's kind of talk about those things, okay? So what is diffusion? Movement of particles from high to low, right? So let's set up a little box or something, okay? That's a membrane, right? So let's say we have particles. We have these particles, and we have these particles. Okay, looking at that diagram, only talking about the particles, what direction will they move? Yeah, they want to move to the right, but what are the only ones that are actually going to get across? The small ones, right? So the blue ones are going to get across, right? And then the red ones, they want to, but they can't, okay? Because they still want to move from high to low. Got that? Okay, that's diffusion. I'm going to skip osmosis for now because I got to talk about that. Facilitated diffusion. What does the word facilitate mean? To help. So what's helping diffusion? It's a protein, right? So if I do the blob like that, that's facilitated. So we have these particles here, high to low, using a protein. That's all facilitated is. When you get into osmosis, then that's a more in-depth discussion. So, ready for osmosis? No, but we gotta do it, right? Okay, osmosis. What are the three types of tonicity? Hypo, hyper, iso, right? Okay. All right, so remember we did that lab where we had the bags of sugar, the sacks of sugar? right? And every bag gained mass, right? Okay, so here's the beaker, right? Here's the sack. Let's say this sack has a 0.2 molar solution of sucrose. Sucrose isn't going to move, but water will. Will the water, and again, this is just pure water right here, okay? Will the water move in or will the water move out? Yeah, obviously it's going to go in, right? Because the water environment is the what? This is hypo, right? That means the sac is hyper, and water always moves from hypo to hyper. Got it? Okay. And then you know, what if I change this to 0.8? Still gonna go, but if I were to compare it, you know, if I had a 0.6, 0.4, 0.2, 0.0, let's assume any of these in different, because this is what you did, are in the bag. Okay, which one of these molarities will gain the most mass? 0.8, because this is the higher concentration of sugar, so the water wants to dilute it the most. Okay, this will have the second most going in, right? This will have the third. Which one will be about equal to the environment? 0. Describe 0 and 0. What would that be called? That would be your ISO, right? So if both are, and so should the bag even gain mass? No, it should stay the same. Okay, and that's that part. So I could give you problems like that. I could give you problems like this. 95% H2O, 80% H2O. It doesn't matter. It could be molarity, it could be percentage. Which one is the hypotonic? 95%, right? Because hypo means fewer solutes. So the water will go out. Okay, you good on those? Because you're gonna have a lot of those. So you gotta be able to identify it. Okay, why do we spray vegetables in the grocery store with water? They absorb the water. So the vegetables are what compared to the water? Vegetables are hyper, the water's the hypo, right? Okay. Okay, anything on osmosis? Good on that? Okay. Then we get into active transport. And all active transport is moving things from low to high with energy, right? And the classic example are these. So what is the go-to pump that you know of? Sodium potassium, the Na plus K plus pump, right? So what do you take in? You take in the potassium and you get rid of the sodium. And just remember, you want bananas. You don't want to eat salt, right? Unless you get your banana smashed in your backpack, then that just makes it for a bad day. So, yeah. So, yeah. Okay. So, lesson learned: don't put your banana in your backpack. Just saying. I don't know who would do that, but.
I really put it in there for 1 period.
Okay, so that's a pump. Co-transport. So co-transport is. Okay, so. And let's say you have a whole bunch of hydrogen ions here. Okay, all right, co-transport. These ions are going to be moved to this side, but look at the concentration. What process is moving these? Active transport, because this is low, this is high, right? So we need ATP to make this happen. So then you get all these hydrogen ions here. Now we have a high concentration of these protons, right? Positive charge. They are then going to naturally diffuse back through from high to low. And when they diffuse through, sucrose hitches a ride and comes in with them, right? What do we call that?
Assisted.
Co-transport.
Co-transport. Co meaning together. Remember I gave the analogy, are you gonna live in an all-male dorm or all-female dorm, or are you gonna live in a co-ed dorm? If you do that, so. Okay? No?
So why are they moving in direction and then moving back out?
Because now we have an imbalance in charges, because now the outside has a net positive charge. The inside, because it lost these hydrogen ions, have a net negative charge. And what do we know about positives and negatives? They're going to want to go in because they're attracted. And it's the high and low concentration. Okay.
And then why does sucrose have?
So sucrose, it's like this is the key to open the door to let the sucrose in. So when they go in, this slips in with it. So if this were a pH environment, if we have a lot of these hydrogen ions here, what environment would this be out here? That would be acidic, right? Because a lot of hydrogen ions means acidic. Very few hydrogen ions, this environment must be, that'd be more basic, right? You can also look at it that way. So, all right. Co-transport, ion gated, it's just moving ions. And then endo and exo, right? So which one means to take in? Endo, right. Two types of endo. You remember them? Phago,
Yes.
P-node. And phago, cellular eating, and P-no, liquid, right? So that's cellular drinking. Get it? Yeah, I mean, come up with something in your brain those easy to remember, right? P.
What are we gonna have to know about this?
What?
What are we gonna have to
Just know the difference. That's all.
Yes.
Like not in a situation.
No, you just have to know about it. And then exocytosis is exocytosis, okay? All right, so did that, we did that. Okay, let's talk about environments. Okay. Yeah, pino and phagocytosis, and then exo is just exo. Okay, ideal environment. All right, so we have a animal cell, we have a plant cell, okay? All right, what kind of environments, using the word tonicity, what kind of environment do our cells like to be? Our cells like to be isotonic. Okay, remember IVs? An IV solution is an isotonic environment, right? You don't want to blow up your cells and die in the hospital by hydrating yourself, right? You want to have an isotonic environment. Plants would not do well in an isotonic environment. That means you're giving them salt water. That would make them shrink, right? What do plants need to survive? They want the environment to be hypo, right? So then what can happen? They can fill up and be this crisp apple, or, you know, the fruit or vegetable that's nice and solid when you eat it, okay? So plants want to be in a hypotonic environment, okay? What is it called if an animal cell bursts? What's the buzzword? Lysis. Lysis, right? Meaning Lysol, right? Hydrolysis, okay? What is it called when a plant cell shrinks? Plasmolysis. Plant shrinkage. Okay? Bursting animal cell. Okay? Bursting animal cell, plant cell shrinkage. What'd you say? It's just my horrible left-handedness. I'm gonna clear it in a second. Okay, okay. And then you have one water potential calculation. Okay, so I'm gonna give you the equation. All you have to do is negative ICRT it, and you have your answer. Okay, so you're gonna be given what these even stand for. Molarity, the constant, 0.083, 31.
You're gonna get it.
31. R, no, I'm sorry. So R is the constant, C is molarity. The only little thing you have to do is you have to convert degrees C into Kelvin. And you're given that too. C plus 273 equals,
Wait, we're doing the ICRT.
you're given the whole thing. You just got to do the math. Yeah,
Just like the lab.
yeah, just like the lab. Okay. R is the constant. C is molarity. Yeah, C is molarity, R is the constant, T is temperature.
Molarity is either 1 or 2. One is
Wait, what's
one or 2.
So, okay, here we go.
If it's sodium, it's 2.
I equals, it's called ionization constant. It's either 1 or 2. And 90% of the time it's 1 unless it's sodium chloride. Sodium chloride is 2. That's all. But everything else is 1. Okay? C is molarity. Okay? R is the constant. Was it 0.0831?
Yeah.
Okay? And then T is temp in Kelvin. Okay? And all you have to do is multiply across. Okay? If your number is negative, which it always will be, what is more hypertonic? -1, -5. -5 is more hypertonic. Hyper, right? The closer to 0, the purer the water is. The more hypotonic it is, the closer you get to 0. Okay? So a bottle of water is 0. Everything else is negative something. Got it? Okay, but it's one problem. Okay? I also threw an extra credit on there too, if you can do the math. It's not bad. I'll show you in a second. Okay, we good on this? Okay, we got 15 minutes. Oops.
Ella said no every time.
Yeah? Okay, so surface area to volume. Okay? So if you had a surface area of 24 and a volume to 3, you want to make that into a number. Okay, so 24 divided by 4 is surface area to volume is 6. Okay, 12 to 3 is 4. Okay, 10 to 2 is 5. Surface area to volume. Surface area to volume. Right?
Yeah,
Which-
I don't remember ever talking about
It was like one slide, and I was trying to get you guys all in. It was like on a Monday. Anyhow, here's all you need to know. The higher the number after dividing is the small, most efficient cell. Okay?
Oh, I remember this now.
So this is the one you want. Okay?
The higher the number,
The more efficient the cell is. Got it? And all you have to do is take your surface area that's given and volume that's given and divide them. And whatever answer
higher
is.
number means smaller. Okay, that's
Yes.
That's like
Yeah. Okay. Now, the extra credits, I'm going to give you a cylinder and a rectangle, and I'm going to have you calculate surface area and volume. I'll give you the math, I'll give you the equations, and if you can come up with the right answer, you get that extra credit one right. I'm even telling you what the
wait,
extra
say
credit
it
is.
again.
I just not do that.
You can skip it. Yeah, it's a weird equation. 2 pi r
Yeah.
something. So I'm going to give you the surface area of a cylinder, the surface area of a rectangle. You figure out which one is what the surface area to volume ratio is.
So can we just guess?
Yeah, it's a multiple choice. Maybe you'll guess. You have a 25% chance. So, okay, and that deals with cell size, right? So cell size. Cells are more efficient because they're small, right? And you want a surface area to volume ratio that when you divide those, you want that to be a higher number. Got it? Okay. Now we'll get into the organelles, okay? Prokaryote versus eukaryote. What is the only example of a prokaryote? Bacteria. Here's the book definition. Prokaryotes lack a nucleus and membrane-bound organelles. Okay. The only organelle they really have are ribosomes, right? And they have one chromosome, and what's the shape of their chromosome? They have circular DNA, okay? Eukaryotes, the definition is they have a nucleus and they have membrane-bound organelles. Eukaryotes have a nucleus and membrane-bound organelles, okay? That's kind of your background. Now let's get into the organelles, okay? All right, so when you talk about this right here, the endomembrane system, what are we making? That's the protein pathway, okay? So, nucleus. Nucleus contains the DNA, right? That's the code for everything. In the nucleus, you have the nucleolus. What's the nucleolus make? Makes ribosomes. That was a quiz question, right? Okay, outside of the nucleus, you have, so here's our nucleus. You have this, okay? And on this, half of it, we have these red dots, okay? I'm gonna put some ribosomes out here too. All right, so nucleus with the nucleolus. There's our DNA, right? Rough ER makes proteins. It's rough because it has what? Ribosomes. These are attached ribosomes. These are free ribosomes. Which type of ribosomes keeps proteins in the cell? Free ribosomes keeps the proteins in the cell. If they're attached, that protein is going to go out of the cell. That's the difference. Because if they're attached, let's go to the next organelle. After that protein is made, it's going to be sent to this organelle. It's going to be sent to the Golgi, the packaging center, right? It ships it in a what? In a vesicle. In a vesicle, right? And then that vesicle fuses with the Golgi, and then it comes out in another vesicle, right? So now we package it, and then that vesicle fuses with the membrane and releases, right, using exocytosis, right? That's exocytosis. Okay, so that's the whole protein pathway, right? So nucleus, nucleolus, now we have smooth ER. Let's do that one. What does smooth ER make?
Lipids.
Smooth ER makes lipids and detoxifies you. So your liver cells have tons of smooth ER, unless you're an alcoholic and you've destroyed it. So, yeah. It makes lipids and detoxifies, right? Smooth ER. Smooth ER. Okay, so we just knocked out the whole endomembrane system. You good on that? Okay. Lysosomes. The word lysis is always a dead giveaway in biology. What do lysosomes do to them? They break stuff down, right? They are the garbage truck of the cell. So they break stuff down. So think of them as sacks of enzymes. Those dots are enzymes, okay? So they break down, for example, food vacuoles, right? They're also involved in getting rid of your webbing. What's that called? Apoptosis. There's also genetic diseases linked to lysosomes. Because what if these enzymes don't work right? What'd you say? Tay-Sachs disease, okay? And that's CHS, not SACKS.
Is Tay-Sachs the only one we really need?
That's the only one you need to know. Right. So Tay-Sachs, you break down lipids in your brain if your lysosomes work right. If you can't break down those lipids, you accumulate those lipids and they crowd out your brain cells and take your life. Yeah, it's a sad disease, right? So that's Tay-Sachs. Okay? All right.
I'm still writing.
Okay. So we're ready for vacuoles? Okay, 3 types of vacuoles. Plants have the big one called central vacuole. It's a central vacuole, primarily stores water, right? Our vacuoles would be like this, and like I said, lysosomes break them down, food vacuoles, okay? And if you're a paramecium, you have this vacuole. The squirter, the contractile vacuoles, right? The squirters. Paramecium. Paramecium, right.
What's the
You ever seen a sea cucumber's defense mechanism?
Wait, what's the paramecium?
Sorry, I got sidetracked. What?
What's the paramecium?
Paramecium's that picture on the very last page on the lab from yesterday. That a lot of you guys said prokaryotic, and I said, no, that's a eukaryote. That's a paramecium. You just have to know it's the classic example of a contractile vacuole. What?
Squirts the water out.
Yeah, only 3.
What's the plants?
I better not. We better stay locked in here. Huh?
Don't they just?
Their anus releases this white milky stuff.
Yeah.
As a defense mechanism. Yeah, but we got 8 minutes. We better not go there. Okay. Okay. Next one. Endosymbiotic theory. Okay. So cell.
Oh, you skipped mitochondria chloroplast.
Oh, okay.
That's a big one to skip.
That is a huge one. Okay, which one's the chloroplast? The green one. Okay, let's talk about chloroplasts. Chloroplasts, photosynthesis, green. They have the stacks of discs called,
Oh,
what's this part? Thylakoid. Okay. Thylakoid. They have their own circular DNA. They have circular DNA just like bacteria. They have their own ribosomes, and when they divide just like bacteria. So the whole idea with mitochondria and chloroplast, they are essentially almost like living things that live in our cells that give us an advantage because they're there, okay? So chloroplasts, made of thylakoids, photosynthesis, they contain ribosomes and circular DNA, and they divide like bacteria. Mitochondria, same deal, right? But they're not green, but we know mitochondria contain the curvy, what's her name? Curvy cristae. Want me to write cristae for you? Cristae. What if someone named their kid cristae?
It's Christian's mom's name.
But is it A-E?
No.
Yeah, it's not cristae. They have their own DNA, they have their own ribosomes, just like bacteria. Which one are found in only animals? Only animals. Which one, trick question, is found in plants? They both are in plants. Okay? Which one, parentheses, ones make ATP? They both make ATP. So they both make ATP. They both are found in plants. Only mitochondria is found in animals. What? Okay. Got it? And I also tied in the endosymbiotic theory. The evidence for the endosymbiotic theory, they have their own DNA, they have their own ribosomes, and they divide like bacteria. Could be an essay question. FRQ. Could be a great FRQ question.
Wait, so the theory is the 3 evidence.
DNA, ribosomes, divided like bacteria. Okay? Okay, moving on. Okay, cytoskeleton. All right, microtubules, they do the most, okay? Microtubules make up those guys. What'd I skip? No, I just said, I just went over that. Right. Cilium flagella, right? Cilium flagella are made of microtubules. If you sliced a psyllium flagella in half and look down in there, there's pairs. You guys remember the numbering? There's a 9+2 arrangement of microtubules. There's 9 around the circle and +2 in the middle. Okay, so if you see a question about 9+2, it's a microtubule and it's psyllium flagella. Okay? So that's microtubules. Microtubules are also pathways for motor proteins to carry things like vesicles, right? Remember motor proteins? And there's your microtubule. They're little pathways to carry stuff. Okay?
So they're pathways for motor proteins.
Yep.
What is this?
Vesicles.
Why is it a smiley?
I was being funny. That's all.
That was so funny.
I mean, yeah. Nah, she is hating on me. So,
Sorry, I had a bad day.
it's a bad day when you put your banana in your backpack. Okay, microfilaments. The only thing you have to know is muscle contraction. That's it. Microfilaments are in your muscle fibers. It slides. That's when you flex the muscles. Got it? Okay, intermediate aren't on the test, so don't worry about it. Okay? Okay, last two things. We got this. All right, plant versus animal. Let's list.
Wait, are psyllium flagella the same?
No,
Cilia is little hairs. Flagella is like a tail. A whip-like tail is the official definition. So,
Well, yeah, but do they do the same thing?
yeah, they're both used for movement. Okay, plant cells. Give me 3 things plants have that animals don't. Chloroplast.
Cell wall.
Wall. Large vacuole. A large central vacuole. Okay? Those are the main things. Animal cells don't. Animal cells have food vacuoles. They don't have cell walls. Kristen, do you have a question or are you stretching?
No.
Okay. And then the last one, SEM and TEM. Super easy. This is on the quiz, right? SEM, scanning electron, 3D image. T, thin slice of an image. Got it? Okay. So when I went over this, I gave you 2 really good hints for your FRQ on Tuesday. Okay, one of them I said, but the other one I kind of showed you some stuff. So that'll help you for Tuesday's FRQ.
How many questions?
One's a long and the other 2 are short. Yeah. So. All right, but tomorrow we get to have a fun day and enjoy.